you look over, and there's a humpback whale right next to you.
Science is for
And
everyone.
so you're like,
Backgrounds, all
oh,
ages, all
okay.
genders, and all walks of life
Tonight
deserve
we
an
have
education.
on
Come be one
Professor
of us.
Josh Benoit joining us from the University of Cincinnati. Josh, thank you so much for coming on with us. Josh is a 2018 Sigma Psi Young Investigator Award winner. He's got groundbreaking research in the physiology and molecular mechanisms that underlie stress tolerance and reproduction in blood feeding arthropods like ticks, bed bugs, tsetse flies, and beyond. He's regularly acknowledged as a University of Cincinnati research rainmaker, which puts him in the top 25% of all campus researchers in sponsored funding. And it's his third time already receiving that honor. He's been multiple times honored by University of Cincinnati in undergraduate mentoring, including in 2014 for outstanding mentorship of student researchers. He leads the lab of insect biology, where his team explores integrative physiology of arthropods, stress tolerance, dormancy, reproduction, metabolism in mosquito species, bedbugs, ticks, fruit flies, and beyond. His research takes him all over the world, including Antarctica, which I'm very excited to chat with you about today. A big study that was published this past year revealed actually that fruit flies are genetically involved to avoid parasites with sacrifice sleep, which we've actually reviewed on stream. Josh has been given over 250 invited talks, won about 50 scientific honors in both research excellence and collaborative leadership, and he's celebrated constantly for mentoring undergraduate research success. Josh brings a very rare combination of molecular depth, lively storytelling, bridging the gap of the microscopic world of insect life while pressing questions about public health, ecology, resilience. So guys, get ready to dive into the science of stress, survival, and insect biology with Dr. Josh Benoit. Josh, welcome the heck on in. Thank you so much for joining us.
Oh, thank you for having me.
One thing we would always like to start off with, Josh, is how you got into the scientific field. What is it that attracted you towards the sciences, and did you always know it was going to be bugs?
Not necessarily really. My mom always says she thought it would be bugs because that's all I did as a child. But I was really set on being a medical doctor and thought that I was going to do that. And then I played football in college, and then I broke my ankle and tore my Achilles, and then started doing research. And it turned out I liked that a lot more. And I was probably better at that than being a doctor because I think I'm better at dealing with insects than I am people some days. And so I just decided that was a life for me, and I started doing insect research when I was probably 19, and now I've probably been at it for about 25 years.
How did you, so with the breadth of insects that you've worked on, where did you start? Which was the first insects that you decided to start studying? And do you remember the research question back then?
Yeah, the first question we started looking at was a little bit of biology, really biology of ticks. And so that's what a lot of my work was as an undergrad and part of grad school. It was really, so they're really weird in how they drink water. They don't really drink water. They just drink water by absorbing it from humid air. So it was kind of working on that process a little bit. And then eventually I kind of moved to mosquitoes, then tsetse flies, and that sort of thing. And so it was basically tick biology and how they don't die when they dry out.
So why don't they die when they dry out? It's what
Well,
was the
it,
end mechanistic takeaway of how do you even, also one thing that our chat usually asks too is how do you
yeah,
work with something, I guess number one, that could be a threat to you, right, in terms of biting you, and then number two, where you don't really have any genetic tools. So how do you do something fundamental without that toolkit that you have in fruit flies?
it's, I mean, nowadays it's getting to the point where the fruit fly toolkit in mice and C. elegans and all those toolkits are great, but nowadays there's a lot of other things you can do. You can suppress some genes. Pretty much every important tick genome's been sequenced. They've gotten CRISPR, like genetic mutants, to work on them now. So there's a lot of kind of tools you could use for those. And how do you work with things that can bite you? You pretty much just watch them really well. And ticks are pretty easy because at least they can't fly. So
Uh,
the worst one to deal with are mosquitoes and CT flies and those sort of things. For the ticks, if you contain them in a nice container, you can usually handle them pretty well. Luckily, it depends on like where you're working. If you're like working on ticks and you collected them from a really like heavy Lyme disease area, you really want to make sure that it actually then you want to be really careful. Other ones we work at are lab colonies that are not as likely to carry a disease. They can bite you, but it's probably just going to be more of an annoyance.
so you can breed these animals in the lab. You don't have to wild catch them every time.
Yeah, you can breed about 10 or 15 different tick species people have been able to breed in the lab.
Nice. Yeah, that's always a hesitant thing on ant species that we talk about here is if they have a mating flight, then what can you really do unless you go out and you actually collect them? But it's cool that you can do both the field
Mm-hmm.
research on the ones that are infected versus the ones that are back in the lab that you know are clean of a disease, but still study the basic genetics behind
Yeah,
them. When
yeah. They're still a real pain because you got to have goats and rabbits and that sort of thing. A lot of ours we can order them in the mail too.
so hold on,
So
hold on, John. What do you
we have I have a tick guy.
you have a tick guy, but hold on. You have to have what do you do with the goats? Like how do you are they on like lab premises? Like for those of us not working with these animal like the ticks, how do you keep how do yeah, from the chat, how do you keep ticks in a lab? Like what is just like
Yeah.
the process
You,
for
you,
that?
you got to have something to blood feed them on. A few people have got it to work artificially, but it doesn't work that well. So if you want to keep them in the lab, you usually got to have some rabbits or that sort of thing to feed them as larvae, and then probably some goats or rabbits to feed them as adults. And you just keep them going, like, and then they live a long time. You feed them and you keep them going, or as someone said, or sheep. Some people use cows as well. And so we usually sometimes only have to do like one feeding stage and don't really worry about keeping a full colony going. And so it's not as big of an issue. But the problem that you really get with them is you can only do usually feed a rabbit with the ticks once, and then they're resistant to the ticks and they won't complete. So you have to get a new rabbit or
Wow.
a new goat. And so if you, for people who do a lot of research on them, that's a real big concern is like how often you have to get new animals.
Presumably that cost also starts building up too when you're trying to study
Mhm.
them. So having the tick guy is a much easier
Yeah,
approach to be able to do.
yeah.
Nice.
For what we need to do with them, a lot of times we get like adult ticks, and we're looking at like maybe how they survive the winter, how they like quest, which is why how ticks search for the kind of a host, and so we can do the studies without having to get them from the lab.
So what was after you had completed some work on ticks, like within graduate school, when did you decide to transition to another insect and or kind of open up that toolbox of looking at new insects?
It kind of just fell in my lap. So I also did for you didn't even mention this. I did a lot of like bed bug research, like through grad school, and those are they're a little hard to deal with. And so pretty much it's usually just been for me. It's more of like what's interesting about the organism, and then to study that. And then I've moved from organism to organism through the years. Probably worked on about maybe 15, 20 different types through the years.
What has been what like the most challenging that you don't currently have that you maybe if everything had like worked out in terms of like I could just snap my finger and it'll be easy for you to have, but like at the time or even now it's just frustrating to keep, but you still have like this back of your mind, I'd really like to still work on this thing.
I would like to work on fleas. The problem is usually a lot of people who work on fleas, you get fleas. So the issue is anybody who like does those, they're pretty hard to keep in a lab because I mean they'll jump 2 3 feet, and I mean they have a pretty high jump. So you'll reach down in there, they'll get all over everything. Their larvae survive really well just eating on like skin on the floor, and so they're really hard to keep contained. And so we had a few for like one little project, and never again, ever. Like I would like to work on them, but it's the idea of I'm pretty sure like my entire department would have fleas if we try to keep them in the lab because they just get out. They find ways. They squeeze through cracks, and before you know it, they've infested the entire like lab.
And I guess their
And
survivability too, like you were saying, on such little amounts of food is really cool. Like really interesting
yeah,
science, but at the same time, that's what allows them to be spreading so quickly throughout the entire department. I'm
yeah.
surprised I would have thought bed bugs too would have been that kind of danger of bringing them home.
Bed bugs don't jump. They don't fly.
So you can see,
So like and they don't like water. So you just pretty much you build like you have one tray, then you have a bigger tray, and you build a water moat, and you got them.
see, that's a
So
really, really cool trick. That's like no one would know that unless you actually work with them.
yeah, yeah, they'll reach and touch the water, and then won't go into it, and so then you pretty much have them contained at that point, and they're easy to work on. So as long as you don't touch when you have your hands working on them, as long as you don't actually touch them, then you have them contained. And so there's even actually some like kind of little plastic pieces that are meant to be put underneath like the stands of your bed that you can put little moats for water or oil, and then they won't come up and crawl onto the bed. So
Oh, all right. That's see, it's one less terrifying thing. I'm sure it's like if you ever, working with someone, be like, all right, I'll be home. I'm working with bed bugs today. You're like, wait a minute. No, 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 it's fine. We got a
yeah,
moat. We got a moat.
yeah. Most of the people the who work on them, they're pretty good at handling them. So
Nice. So,
yeah.
which insects did you decide to settle on for working for your lab, and how did you make those decisions? Like, what was it about the insects you have now that makes them attractive to study and that you get excited about learning about them?
So right now, it's mainly been about a little bit about the question. So mainly we work probably on mosquitoes and a wide variety of topics within mosquitoes. Mosquitoes aren't necessarily that hard to keep in the lab. So they need a blood meal every couple months to lay eggs, but they're relatively easy compared to some other things. We have those in kissing bugs and then ticks too, and then a little bit of work on Drosophila. We don't really keep the Drosophila in the lab. Another group rears them for us. We just do the experiment. So it's mainly some ticks and then kissing bugs and then mosquitoes, mainly because we like they're relatively easy to rear. We have a couple students or staff working on their projects, and really they all have interesting aspects of their biology that we're really interested in working on. And so it's just a combination of all those factors kind of allowed us to kind of settle on those. And it's just the more topic areas and more organisms you have, the more you have to read and keep up on the literature for those. And so maybe at some point I'll cut back to like maybe one or just two. But I think right now we have like four or five different kind of organisms we have in the lab that we're doing really some studies
What are you feeding the mosquitoes?
on. Either we have an artificial feeder or we have a few that are live host feeds.
And how do you decide like which one, just from the feeding perspective, which one you do? Because I know that some labs have like you put in your arm, other labs you put in a bunny, other labs you have the artificial.
Yeah. We have two species that just will not feed artificially well enough, and so they get a live person host, and they'll feed on them, and then the three others that we have mainly they'll feed artificially. So we can just give them an artificial blood meal, and then we can do enough work with them. It's really two different the two different ones, and they don't need fed that often, and only for like maybe every 6 weeks somebody in the lab feeds them, and so then they lay eggs, and then we keep them going, and then we only end up blood feeding them for like experiments if it really necessitates it. So like every so often we're interested, like, oh, we actually do have to blood feed these ones on a live host because we're interested in like this specific thing, and we don't want our like artificial feeding to mess it up.
Right. So it's depending on what question that you're asking and just adapting the feeding best to answer that.
Yeah. Yep.
Nice. For so one thing we kind of wanted to do was kind of a whirlwind of all the different insects that you study and kind of your favorite stories on what you've studied on them in terms of research question and how you were able to answer those kinds of research questions because
Okay.
the community's got a lot of questions on just the non-model organisms and just the
Yeah.
variety of insects you've worked on. And I was just wondering if we could start off maybe with the cockroach story and them giving birth to live young. Like what pulled you to that question and
Yeah.
how did you go about studying like
So
cockroaches?
kind of goes back to my tsetse fly days. So tsetse flies also give live birth. So the reason I got interested in that was my PhD advisor worked on that when he was like in his early 30s. We didn't work on it in my lab during my PhD, but he was always talking about how he enjoyed this system and that. Then an opportunity came, and then I was out at Yale for 3 or 4 years working on live birth and tsetse flies. CT flies are a real pain in the butt to keep in the lab. You got to feed them blood 3 days a week. It has to be like pristine blood. Like you can't accidentally get any bacteria in it at all. They die. And so it's like they're very difficult to keep if you want to make them your main focus. So then I decided, well, I was really interested in doing live birth stuff, and I knew there was this extra cockroach, this Diploptera punctata, which is a live birth cockroach. And so then eventually I started moving towards that, and then started working. I had a grad student, Emily Jennings, who was really interested in working in that area. And so then we started working and eventually did some studies on that. We actually have a new study coming out that'll actually be in the Journal of Experimental Biology that should be published probably within the week about the dynamics between sleep and pregnancy in this cockroach
Oh,
and
congrats.
how if they don't sleep correctly, the pregnancy cycle gets extended.
Interesting. See, we I'm always been a big fan of your work, so we usually review the papers whenever you publish or put up a preprint on stream. So we're
Yeah.
looking forward to seeing that one.
Yeah, I think that one officially the uncorrected proofs is probably on already on Journal of Experimental Biology, but the journal is actually going to highlight it. So
Perfect.
it'll actually be, I think it should be officially published by mid-August.
Nice.
So, and
Congratulations.
to do a quick shout out to that, that entire project was more or less undergraduate led by an undergraduate named Rania Freegard. And so she's now working at the NIH and then going to med school next year. And so she was responsible for a big proportion of that project. So it was really fun, and I'm glad to see it's actually being highlighted. Like they're interviewing her as an early career researcher as well.
Oh, that's
So
awesome.
it ended up being a more exciting paper than what I thought it would be.
Can you give us any of the highlights as to why that really got you excited?
For that paper or just
Yeah.
the cockroach in general? Mainly, it was initially we were just really interested in like, all right, do they sleep? Like, does pregnancy impact their activity? And it turned out it looks like it makes them less more risk adverse. So like they're less likely to come out and forage for food. And so that's the big thing, the big change in them is that it's actually they end up like less likely to when the kind of the sun sets, they're kind of more hesitant and they come out in shorter little bursts than from their hiding spot. And so that's why like they just and we found that interesting, and so then it ended up being like a cool observations about how they behave a lot different than the non-pregnant females and then the males. So it was a really cool project over a course of about 3 years.
Is there any direct correlation with that to human research? Like one thing that we always have questions about is there a connection to human health with the research question? I mean, and if
Yeah,
there's not, that's great. It's just you're studying it because it's cool, but for the live birth, that kind of brings up imaging of humans. Is there a direct connection when studying that?
a little bit. So a year or two ago, we actually published the genome of the cockroach as well, and it turns out a lot of the genomic changes are very similar to the vertebrate genomic changes to live birth. Like it's mainly to get more oxygen to the developing cockroach fetus or embryo, whatever you want to call it, and then to allow like changes in blood flow and that sort of thing. So some of the aspects you would expect. And so like we saw a lot of different aspects with that. And then right now I actually have a project, and it's another co-author of the cockroach sleep paper, Sinead English. And so we have a big collaborative project where we're actually looking at like climate change in live birth, and it branches viviparous vertebrate and invertebrate organisms using Diploptera as a model for invertebrate organisms. So there's a lot of similarities. It's just the biggest major difference is just the ectotherm versus endotherm aspect. And so it's all insects or just endotherm, so they're relying on the external environment and have no regulation.
Is it easy to keep them in the lab, these cockroaches, even though they're giving live birth? Like, is there any kind of special consideration you have to give them?
No, they just get handfuls of dog food and some water, and we get so many of them that there's just too many, and so they're just in these plastic bins that are about 1 foot, 1 foot by 1 foot, and then they end up just what's the word? They grow to the point we have too many, and then we give them to other colleagues who need them and that sort of thing. But then otherwise, it's they're cockroaches.
So they
You can
still
pretty
like
much
the
get them.
really good survivability that they're known for,
Yeah,
that's going to survive
work
the
for them a
nuclear
lot.
fallout and everything. They'll be around for the lab. That's
Yeah,
a really really cool study, especially on sleep and the change of
yeah.
that. One question that again we get pretty common here is studying insect sleep, right? So there's always different definitions of what sleep is between different organisms. How does
So it's looking more and more like the general one, like you have like changes in neuronal activity, like that sort of stuff. Like certain areas are suppressed, other areas are increased. There's like for at least in insects for a long time there was like specific postures. There's times when it should be higher, and there's a few other categories like maybe like respiration metabolism's down. But now there are some recent cool studies tracking eyes and spiders, then the show like that it changes when they're under their sleep like states, the movements. So and then been some recent neuronal recordings of some Diptera species that show that it's likely the brain signalings change when they're in their sleep states. So it's looking more and more like the differences in how you call sleep between the different organisms. Like maybe insects don't dream, but there's people who don't dream either. So it's like I'm not trying to be philosophical with that, but like they actually just don't remember their dreams, or they just really just aren't. And so it's maybe it's not as different as they once thought.
Yeah, that was actually a question from the chat is that do they dream? If there's already, all these neuronal signaling changes and chemical signaling changes, is there a possibility that there's also some kind of dreaming, or is it memory consolidation? And I guess is that different from dreaming?
I don't know. And so it could be. Sorry, I'm yawning. It's been a long day. It's been a little bit of potentially they could, but until someone actually can validate that it's actually happening, it's like I mean like most vertebrates will dream when they sleep. So like your cat dreams, your dog dreams, mice dream, like that sort of thing. But then whether or not it happens in insects, who knows? I mean, mainly if you're getting these changes in kind of like the neuronal signaling, is it a dream or is it just like when you turn it off, there's repairs going in one part that's allowing you to like synthesize what happened in the day and potentially like have longer term memories from it. So, but whether or not it's actually dreaming is a little bit is a little hard to tell.
Do you know if when doing these recordings from the chat, is there a REM sleep equivalent in these animals, or is it when you're defining sleep, it's just kind of the on or off switch?
I believe the most vertebrates usually do have some sort of like REM type sleep as well. Like they have the lighter sleep and then the deeper REM sleep like humans have. So,
Cool. What's coming up next for the cockroach research that is really exciting to you?
oh, what do we have? Well, we have a couple studies. So the with I can talk about a little bit. I'm trying to think of which ones I can talk about. I'll talk about the drought stuff. So we have some really cool studies about how drought impacts pregnancy. It turns out the pregnant females can like not drink water for 2 weeks,
Wow.
and then the progeny survive. And so we have some a project on the mechanisms of why that happens. And so that would probably be the most interesting next step. We also have one looking at immunity and other factors associated with pregnant non-pregnant comparison. So that's kind of like the projects that we have moving along. But like within those, and those are kind of moving along slowly. And the only reason they're moving along slowly is I don't have a grad student or someone devoted to that area right now. So it's both of the projects are undergraduate led, which by me, and so which means like we make slow progress, and then all of a sudden when we finish it, then I help write the paper, but it's not like there's like a big push like to get it done.
So it almost
With
seems
that,
like with this model, you're able to ask a lot of different environmental pressures, right? And how
mm,
they affect different, like the pregnancy itself. And then there's a direct connection that you can make to other vertebrate models, even with
mm.
respect to that live birth. Is there a perturbation that you'd be really interested in studying that maybe haven't hit yet?
We're doing some stuff with some like artificial light at night with it.
Oh, that's really cool.
Yeah, and so that'll be those are we're pretty early in those studies right now, but it looks like it's yielding some interesting results. So we'll probably my guess is we'll probably have most of that done by probably Christmas because it's a lot of tracking, coding, that sort of thing, and it's pretty easy to actually do, and so it's a lot of setup. Let them run for 2 weeks, and then we get a lot of data from it. But then it'll probably take a year to get the paper done.
Can you comment a
So
little bit, Josh? You were saying that there's a lot of and we said it too when you came on that you have a lot of undergrads working on these projects.
yeah,
Um, can you just highlight it for the chat too, like why that's really special? Like I think we all like the community here really appreciates education and bringing in people and not gatekeeping, which is one of the
mhm,
reasons I wanted you on the show. But just
yeah.
for maybe if someone's passing by or listening to this later, of just some of the importance behind that.
So it's pretty typical. Most labs don't have that many undergrads working in a lab. Usually we have 15, which is a lot. But like, so a lot of times within my lab, I can maybe directly supervise 2 or 3 undergrads. And my direct supervision of undergrads usually focuses on the area of the lab that I want to keep going, but I have no grad student or postdoc working in that area. And so right now it's the live birth. And to be honest, I love the research. I love the area, and we're still moving stuff along. I have my really good colleague Sinead at Bristol, and I like working with her. And I like the projects we're doing. So if it wasn't for like her still going with stuff and the projects, I probably would have stepped back from it. But I actually do like it a lot. Like I like the group, I like the meetings, I have with them. So like so I'm like, all right, well, I'll keep this going, and I have that work, and then but then a lot of times, so I have like in the lab. So I can say one of my grad students, Saida. So she's doing work with kissing bugs. So she has 2 or 3 undergrads who work with her just because they want the research experience, they want to do the work and that sort of stuff. And then she also needs to learn to supervise people and have her own like little group. And then it also allows us to get more done. A lot of the science projects is just you got to do 30 replicates sometimes, and you may be able to see like oh, hell, sometimes you may have to do 100, and so and you may be able to see like all right, you see this is happening after like 15, but you're like yeah, I got to do it 40 like 30 more times, and so you get undergrads involved. You know how to do it. You show them how to do it, and then they can independently do it. And then by getting 3, 4, 5 undergrads along with the project, it can free up a grad student's time to maybe do a little bit more like supervision roles or writing or developing the project. And it's pretty common. Like yeah, like I said, like you did research as an undergrad. What was there? Maybe two in your lab,
Yeah.
two undergrads maybe. And so usually on a given year, we have 15 to 20, and we try to grab them as freshmen because we try to grab them so early, and we try to get 5 or 6 new freshmen each year. And our real hope is that they stay around for 4 years or 5 years. And if you stay around for 4 years or 5 years, then they can end up getting on like multiple papers by the end. So that's really why we like to actually have the undergraduates involved.
I mean, that's an immense investment into the future. So as that's,
Yeah,
thank you for doing that because it's not something that most people go after and are excited about and then brag about even. Like when you, right, that's the first paper you told us about is on undergrad led project. And I think that just really
yeah.
speaks to the investment you have in the next generation of scientists.
And we've had through the years now, I think we've had maybe 4 or 5 full undergrads. So like the Antarctic Midge, the big reproduction paper, that was led by an undergrad, Jeff Finch, who is actually just about to finish his PhD at Arizona on plant genomics. And so he completely switched. But all right, yeah, great. But like he was great around the lab like 2 or 3 years. And then we've had undergraduates go into stay in the lab, go into grad school. And so since I think now I've had 93 that have gone on to
Wow.
med school or dentistry school through the years now. So, and then like a few, like probably like 20 or 30 that have gone to grad school. So it's just, there's just a lot of stuff that they can do. And like I said, like usually when they come in, they still got to kind of like cut their teeth and deal with the crap for a little bit at the beginning because a lot of like when you first start and it really like it's a lot of like all right we got to set up these cages and that sort of thing, and so it's going to take a lot of kind of like boring work, but the really good ones who are really interested in it they kind of shine to the top and very quickly like take over their own projects, and they may even have 3 or 4 undergrads who work just for them. And we've had that happen. Like they're seniors and they have a couple freshmen, sophomore undergrads who are working with them. Then we just tell them what to do. So I have an example of that of a person right now, Gabby Lefevre in the lab, and she's doing stuff. And so she's been around 3, 4 years. So rather than me telling her exactly what to do, I say, hey, here are these samples. We need to do this. You got it. Just give me the data and like a few months. And so then she has another undergrad who's helping her out. So they meet, go over and do the experiments, and then they come in with a stack of the in their notebook with the data, and then it's already put in usually Excel. Then they've already made preliminary figures in R. They've run the stats and say, hey, what do we need to do next? And so rather than me having to even meet with them every week, I talk to them maybe once a month, and they just kind of are on their own. And so like that's the situation we really like to see.
Yeah, that's an incredible situation to be able to come back like, all right, professor,
Yeah.
here's the data. I've got it analyzed, ready to go. What I think the next step would be. I'm like, whoa, that's
Yeah.
really, really impressive.
And sometimes it's just you gotta do it once more. And so not even anything's wrong with it. It may be like we got a data set from them, and I looked at the data, and we're just validating a previous data set, and the last data set we had a few more, and this one we had like 30, and it was like a little tiny bit from being significant, but and we thought we may need to do more just because, as you know, it's like a little bit of behavioral physiology data, and there's just a high level of variability in that. So to actually get a significance, sometimes you have to run 60 individuals, sometimes 90, and it is what it is.
It's a little squishy because of just individual variation, but the trend is
Yeah.
for sure there.
Yeah, or like this individual mite that we were interested in, it turned out it ate something right before the assay, which the other ones don't. And so you're like, man. So then you just have to do it a few times.
Right. So you mentioned in the Arctic midge. Would you mind
Mhm.
going into that story? How did you get into something like that? Did you go to the Arctic and research this animal? How does one even study it? Josh, we want to know all of
Yeah.
it.
So the Antarctic stuff was probably one of my fun time of life. I would love to go back again. It's hard to get grant funding, just in general. It's hard to get grant funding to go to Antarctica. It's really hard to do it right now. So we were really interested in this midge because it can freeze completely. So it overwinters by freezing, and so just frozen, and that's it. And so I got lucky when I went to grad school. My PhD advisor had a grant to go down there. That was one of the reasons why he lured me. I was really insistent in leaving Ohio. And for those of you who don't know geography, I'm in Cincinnati, which is in Ohio. It turned out I moved back later, and so but I was after undergrad, I was interested in going somewhere else, like moving, that sort of thing. So I decided that, I interviewed at like multiple places, and I had a few offers, and I was really interested in them. And then eventually, someone was like, "Oh, you should go meet Dave at Ohio State." And I went up there, and he said, "Well, I heard one of your big things is that you want to get out of the country. So if you come here, I will send you to Antarctica at least once, and then Africa for research at least once." And that's the thing I can throw out there. I'm like, all right, let me think about it. And so then I went and started thinking about it. And then he also said, I don't really care what you're going to do, work on. Mainly, you find a line, it fits within the grants, and I'm just going to let you go. And I started 10 days after I finished my undergrad, walked into the lab, and just started doing work. And he's like, well, I got one project I want to do while I'm thinking of what I want to do for my PhD. Did that project, and then developed my PhD project, and just went.
Wow.
And and then I got really lucky. I got to go to Antarctica twice rather than just once. Actually, I've been three times, and so I got to go. My first winter in 2006, I go in, and it turned out one guy who was slated to go got a really good job with the USDA, and they tried to see if he could be delayed and go after it, and they're like, no, like you have to like start by November. So he couldn't go. So they said, well, who else should we go? And they're like, it's not easy work, and it's actually pretty like on a day-to-day aspect. The sun's up 18 hours a day. You're working 16 hours a day while you're there. So it's not like, I mean, it's a lot of work. So then I say, hey, do you want to go? And I'm like, yes, I want to go to Antarctica. And so I went and enjoyed it and did the work, got a few papers. There was another trip coming up the next year, and they're like, hey, like, do you want to go again? And keep in mind, none of my Antarctica work was in my PhD at all.
So what does that 16-hour day look like? And I guess too, how long did you end up going?
I went total. I've been down there for about a year total through time. So I think it was like actually a little less. I think it was like 2 months, 2 months, and like four. So about eight months
Mm.
total down there. Then I down there. You get up, the sun's up, and it's like 6 a.m. and you're having breakfast, and then you take a boat out to go to the little islands, collect samples. You come back, process the samples, and then you start experiments all afternoon, and you're maybe running them, and sometimes you got to run them 24 hours on certain time frames, and then maybe you're not going out the next day, but in it's a lot of work, and then most of the staff would get Sunday off. But as a scientist, when you're down there, you don't usually. Like, I think my entire time down there, I think I probably never took a full day off. So there was always something to do. There was a few days, like maybe it's like a Sunday, the day where everybody has off, where I'm only allowed for like 4 hours. And maybe they're like having a movie night or like a special dinner or something, but then in general, it was a very different situation. And so, yeah, and so people saying it was a incredible opportunity, and it was an incredible opportunity. I would not trade it for anything. It was, I would love to go back again. Then I went back in 2017 or 18 as faculty at that point. That was a lot harder I was a lot older, and then also I had my own lab that wasn't really focused on Antarctic midge research. So, but I was still doing work and got a few papers out, and that was where like Jeff and that paper came out. So that was like adding extra work on top of my work, and then I still had to have like meetings with grad students on the phone. So I'd call and have like like talks with them once a week, and I'm like working like 12, 13, 14-hour days and having like meetings with them like midday to talk about projects, that sort of thing. And it was just a lot of time. So yeah. And
Uh,
so, but like I still would, if I could get my own grant and do some work with it, I would love to do that again. Take some students down with me. I love, you always either go to the bottom of Argentina or the bottom of Chile. So either Ushuaia, Punta Arenas, and I love those towns. Are great little places. And I would like, I have ideas for projects, but whether or not I get a chance to go back again will be like, I'm assuming it. I probably won't. So
Before we chat about like why the research on the Antarctic midge excites you, we have a
yeah,
few questions from the chat of just like
sure.
what goes into in terms of like a medical to be able to go down. And then
Yeah,
I guess too like if you were to go down now and take students, how easy is it to take students as well? And then how cold does it get? So like what does the research lab that you end up going to look like? You know, is it in an igloo? Is it
yeah,
in a building? Like what's it even look
yeah.
like?
No, it's there. There's three main U.S. bases there. There is um, South Pole, which is that's South Pole. Like you're in the middle of Antarctica, you're on ice, and like when you think of like, then there's Palmer Station and McMurdo. McMurdo is the biggest base. It's you go through there through New Zealand. And so that's the biggest base. A lot of countries share and use that kind of as their base moving around. The one I was at was Palmer. Palmer's on the Antarctic Peninsula. The best I can explain the weather while I was down there is I went from Ohio winter to Antarctic summer, and it was more or less the same exact thing. It was like, and so it was like kind of cold some days. Other days it was like above freezing enough that the midges were out moving around. So it was like, and I would even make it a little bit warmer, maybe like Kentucky, Tennessee
Oh
winter.
wow.
So but that's the summer there.
Yeah.
And the weather, and they say Ohio winters can be crazy on the chat. They were crazy. It's like there you're on the coast. All of a sudden you would have a day where it may be like almost 50 degrees Fahrenheit. The next day would be below freezing. And so there's just a lot of variability. And where we were at in Antarctica, they don't use sled dogs anymore. They got rid of those. It's either snowmobiles or, well, the spot we were at, we use boats to get around because it's coastal. So a lot of the research on different islands, you take these little Zodiac boats, use it from island to island. And that is its own adventure because you'll be zipping along, then all of a sudden you look over and there's a humpback whale right next to you. And so you're like, oh, okay. And so there's pictures of me with this humpback whale swimming right along our boat when we were just going to our little site to collect insects. So it's actually like, I mean, it's a great, it's a great research. And again, like the entire idea was just really figuring out like different population of these midges and then how they survive in this inhospitable environment. And a lot of the work is really how they can freeze completely and just come back to life.
So
So
you have to do that work on site, I take it. Like you're saying, like your 16-hour days, you're going back to the lab after collecting the samples.
yeah.
Why is it that you can't just bring them back and study them
So
at home?
you can bring them back. We actually bring a whole bunch back. You can bring back the Antarctic midges, but then you're on a ticking clock once you have them back. So you have them back, and then and you also like the way you have to pack them and that sort of stuff. You lose any sort of island variability. You try to keep that, but once you kind of put them under those like shipping conditions, they're just a Antarctic midges, Belgica, just in general. So you can do some basic physiology, some experiments, that sort of thing. So we brought a whole bunch back and actually then tracked them and waited and then warmed them up a little bit and had them pupate as adults to understand their reproduction. We did a lot of that work here at Cincinnati, and you have to get permits to bring them back. And did um, we did that sort of thing, and yeah. But then no one's been able to really get them to reproduce in the lab. So like once you bring them there, you bring back some like plants they like to eat, and you usually got about 6 months to
Hmm.
work on them, and they're usually then gone by that point. You've done all your little experiments, and that's it. Like we still have 3 or 4 experiments left to publish that we've just never even gotten to publishing, just because someone has to write the papers, and that's in. If you've never talked about it on here before, the probably the biggest speed bump or delay in finishing anything is finish writing the paper and doing the data analysis at the very end, because you can sell the data, but then someone has to spend 70, 80, 90, 150 hours if it's like a first paper writing up like a report of it, and until you have someone to do that, it usually just is like stuck there.
Yeah, it's we've definitely highlighted a lot the publication process, the difficulties, frustrations, and beyond, and the beauty of preprints too, which is why we always appreciate your papers because they're usually preprints and we can actually all access them and talk about them.
I put everything, since it became more kind of acceptable, up as a preprint. So we have a couple more papers we'll put up as preprint soon, and then the ones that are not are usually probably like maybe like a review article, and so they're not the primary article. And you're not really supposed to put reviews up as preprints because there's no reason, because it's not new, unless it's like a synthesis review, so where you're actually doing new analysis with it. But yeah, usually all every new article we put up as a preprint, and I encourage everybody to do it. And nowadays, all the journals are fine with that, because it's from like we've had a lot of papers. So like I have a really interesting paper on like mosquito egg thermal tolerance, and you can go find the preprint and then find the final article to see how much it changed
Yep.
due to the reviews. And so it's more or less not even the same paper anymore.
Yeah, but it's cool. Like you get to, it's two for the price of one. You get to analyze it twice.
Yeah, yes, analyze it twice. That's what we
Yeah.
can call
Well,
it.
for us here, we excitedly analyze it on the show.
Yeah.
We had one more question on the Arctic midge story. Is number one, I guess, why does one study? Like what can we learn from the Arctic midges? And two, what are some outstanding questions that you'd be really interested in learning about with these animals?
We're really interested, like how like there's some pretty cool, interesting data about how the specific microenvironments and nutrients impact them. I'm really interested in that. The main reason that comes down to like why it's so important, mainly they're just a limited species, but it really comes back to that full freeze tolerance. There's very few organisms that are fully freeze tolerant, and this is one of them. And so that's one of the reasons why. Like, I mean, if we could ever figure out exactly why, like they actually could survive and everyone will survive, then we could just freeze ourselves. And like say you get some terminal cancer, rather than trying to like wait it out, you could decide, oh, I'm going to go into this like kind of freezing period, and then when they have a cure, they wake you back up. And so that's the sort of thing. Or you could do it with organs. And so like mainly like say people who need organ transplant, they're limited. So sometimes like you may need like a heart, and someone may die in California, but they need the heart in like France, and like you may not be able to get it there in time. And so, but if you could actually, if you figure out the right conditions that you could freeze it, then you could actually just freeze it, nail it, thaw it out, and still have the viable tissue. But like that's still like a pretty difficult process to do, and they've been still trying to settle on that for years and years.
How long can you freeze these midges? Like, is that something that you could just bring back and keep in your -20 freezer indefinitely, or is there like a limited shelf life?
So that's a problem. Like we know a lot of little factors. So it's like some little sugars and that sort of thing that kind of regulate the process. But it's hard to get the conditions right. And that's the same thing with tissues. Is hypothetically there's been a lot of progress towards it. Like, oh, you could do these like use these specific chemical treatments, and get it to like 80% viability. That's not good enough. Like it's still, when you dethaw, you're still going to have 20% cell death, and you're going to die from that,
Right.
or it's not going to be viable. So it's still not, it's not quite where they need it to be.
Interesting. So there's something about the environment in
Mm.
Antarctica when they're freezing
Yeah,
that is like
it,
a
it's
perfect
probably
mix.
it's probably they choose, and so they make a choice to like crawl to a specific spot that optimizes the conditions that they're going to overwinter. And normally, if it was just a random spot, maybe there's 5% survival.
Hmm.
But in this specific spot under this rock, low oxygen availability, they've eaten the right things, they purge their guts, so there's no ice nucleated agents to freeze them. Then all those conditions come together. It's when like the sun's disappearing, and then you put it all together, and then all of a sudden 60-70% survive. And so like that's kind of where it's probably would need to be.
It's you said 60 to 70% of these
Yeah,
actually survive.
will still make it through. Like, and I'm just guessing on those numbers, but like, still not. I don't think all of them are still making it through the winter.
Gotcha.
And that happens with insects here, even the ones that go overwinter or dormancy. Not all of them make it, but enough of them make it to seed the next year.
And that's what people in the chat are, hypothesizing of like humidity maybe influencing it as well, like of what it's in Antarctica and just like where they're crawling and the diet beforehand.
Yeah,
And like you said, the gut clearing makes sense too. Like there's all these extra factors going in that if you can't control, you're not
yeah,
going to be able to have happen.
yeah. And like, say if we just take the midge randomly, pull it out of his food source, and then try to freeze it, it's not gotten itself ready for that
Right.
yet. And so then the survival may only be 25%, where if it had the right environmental signals to start that process, it would have been much higher survival.
So is your holy grail question for this then is just like how exactly mechanistically they're doing it and then replicating it in, let's say, a tissue that's non-midge?
Yeah, yeah. I
Yep.
mean that's a holy grail for pretty much a lot of the cryobiologists is, all right, if we get to the point where we can freeze anything, then we can save everything.
Yeah. It's a
Yeah,
the chat dude like that's a really cool way of framing the research question and just like the massive implications in order
yeah,
to be able to answer that. And
yeah,
speaking of massive implications, the next insect you've got is mosquitoes.
yes.
So Josh, could you tell us a little bit about which species, why those species, and just some of like the coolest research questions that you're
Yeah.
answering right now with them?
So with the mosquitoes, are probably we have a few different species. We have about 7 or 8. It's not really worth going to them. They're all important. Either malaria or viral vectors or one of those ones. They're all medically important, the species we have. So they're that sort of thing. And the two coolest projects we have going on right now, and probably three coolest ones, is we have one looking at their sleep profiles and how sleep impacts their biology. And so we had a study a few years ago in Journal of Experimental Biology on their sleep. We have a few other ones. We're working on one right now on their sleep profiles of like the male mosquitoes as well. And so it's how proper sleep cycles and their circadian rhythms or daily rhythms, if you want to call it that, impact their behavior in blood feeding and disease transmission. So we have a couple papers out already on that, and then we were working a few more. So that's a really interesting area. And then the next one is not just mosquitoes. We're looking at how humidity regulates circadian rhythms, and so that's an area we've been working on. Then the last one is a recent area. In the biology of things, you have little tRNAs, and they're responsible for taking the amino acids and turning your mRNAs into actual proteins in organisms. So tRNAs, people just assume like an AAT may code this specific amino acid, but it turns out they're much more complicated than that. They have a whole bunch of modifications that make them function properly. So we've been looking at how these modifications are important to mosquito biology. And we have a few recent papers that show how like pools of tRNAs and their modifications vary with like blood feeding and reproduction. So those are the kind of the main areas we've been working on recently.
When studying the effects of disease state on them, like about like, how the sleep pattern changes and blood meals, do you
Mm.
have to infect them with a particular disease to be able to study it, or do you feel them in a cured state is a sufficient proxy.
It depends. So there's a lot of different ones, like malaria, like the arboviruses or vectors. They have a lot of impacts on the mosquitoes. So then there's some ones where like the rhythms may change and those aspects. Right now, a lot of the sleep profile stuff, we're just focusing on the unfed ones because we know so little about it that just even like doing the basic assays of those and understanding before, because the mosquito having the disease isn't always is critical. The mosquito not having disease and understanding its behavior is critical as with it having the disease, because it still has to pick it up, and so it has to go bite something and get the virus or the malaria parasite that, and then bite somebody else. So understanding what happens on that first feeding is important to disease transmission as well.
So the field is so new that even these basic what feels like a basic question of just like what is their base behavior on these is pretty it's still unknown.
Yeah, I mean, not their base behavior. There's a lot of known about mosquito behavior, blood feeding, host preferences, like how they locate the host. I would say for insects, the order of like study is Drosophila, and let's just say Drosophila melanogaster, then mosquitoes. But it's probably Drosophila up here, then mosquitoes right here, and then a big drop off after that. Maybe Tribolium comes up. So some like specific beetles comes up way after that. But in general, it's probably mosquitoes are probably the most studied insect outside of Drosophila, just because Drosophila, you got your little itty bitty jar, and you can have a entire 90 populations that sit just on your desk. And so like mosquitoes, you can't really do that. Like in that same amount of space, you can maybe have one mosquito lineage or like one cage of mosquitoes. So it's a scaling aspect, but it's that among mosquitoes, I mean, they're probably the most studied other secondary insect besides Drosophila.
Is that something like how much for the folks at home, just how much space does it take in order to actually grow these mosquitoes and like what does a lab even keeping look like on that?
So we have a insectary that has double doors, that sort of stuff to hold the mosquitoes in. We have a cage, and in the cage we have like we have one cage per species. Some of them have bigger cages. So we have big colony cages, which are 2 feet by 2 feet, which keep a lot of mosquitoes in there. And we use those to generate the eggs that we do experiments on. And then we do experiments on the mosquitoes in 1 foot by 1 foot cages. And so then, depending on how many experiments we go, our entire insectary is just full of
Yeah.
cages. And so then we have that. And then that's what we have for the uninfected ones. We have a quarantine facility that has infected insects in it, but that is like on the other side of the building, 4 floors down, and they're separated on purpose. So that's where we keep like those.
Yeah, very cool. And I remember we had talked years ago too about testing different molecules as well to be able to,
Mm.
as effectors to keep mosquitoes away. Could you talk a
Yeah.
little bit about just if y'all are still doing that or like how does one set up the study? Because I remember when we chatted, I've told this story on stream too, where you have a room and you just like letting them out in the room and just testing them in the room. And it was like, remember that old abandoned building
Yeah.
that
Oh,
y'all
we,
got?
we, we
Uh,
still use it. I think they're finally going to tear it down next year. So mainly we do some studies where we want to understand like what the mosquito behavior is on a larger scale. So we have like room size cages where we'll put like an artificial feeder in there, and then we'll put a repellent on it versus one without a repellent, the different aspects that you can do that, and then see how the mosquito, does it actually prevent mosquito from coming over and biting. And so, or we test traps sometimes, like new mosquito traps, that sort of thing.
That's yeah, it's really cool. Like I was telling the folks at home too, you just have the building, you're
Yeah.
just using the rooms, and it's like, all right, when we're
Yeah.
done with the room, we move on to the next one.
Yeah.
I guess one final maybe is there another insect that you're really excited on working on? Either we can go back to like the tsetse fly, which I know that we kind of touched on at
Yeah.
the moment, but is there something in the lab today that we haven't yet touched on that's just like a really cool animal that you've been having
Oh,
study?
yeah. I could talk about. I mean, we talked a little about the ticks, but the one thing that we have in the lab that we don't really have any actual projects going on. Well, we have a paper we're working on it. We work on a little bit on thrips. So if you don't know what thrips are maybe one of the most important crop pests. And so it's also the one that took me my writing the most time to actually fix because it's either one thrips or 15 thrips. So it has an S no matter what. So yeah, so it was pretty hard. But they're a disease. They're a transmitter of tomato spotted wilt virus, which is a really important virus that a lot of times if you get kind of wilty tomato plants or wilty plants and it's spotted, it's a pretty common response. And they think it causes multiple $1 billion in damage. And so, no, the thrips are not no-see-ums, but they're no-see-ums for plants. So they're these little tiny green insects. And I'm guessing if you go to your tomato plants and shake some into like a paper, you may very well see some on there. And so we have a couple projects working on those. We published them. Had a grad student named Sam Bailey who worked on them for a couple years, and so we have a couple more papers left. And I would like to do more work on them, but they're very tiny, and we rear them on green beans we get from Kroger's.
Oh, wow.
And so it's we have them. We just don't have any active projects with them right now. And I have ideas for about 7 or 8 projects, but I just don't really have anybody to do it. And so like we have them in these little jars with green beans, and we just keep them going.
And is it because they're so small, is it difficult to do any kind of genetic perturbation on them? Like, are you just left with behavioral and
No, no,
stress changes, or can you like do
they're-
injections on them?
no, there's a lot of people who do a lot of different work and projects on them and interacts with plants and that sort of thing. It's because they're such an important crop pest. And we did the genome a few years ago, so the genome's been done on a few species of these. So that you can work on them, and I think someone has done CRISPR on them now, if I remember correctly. So at least you can do genetic manipulation. I believe so. Yes, yes, they have.
Nice.
And so so you can do the work on them, but it's just they're they're tiny, hard to keep. They have a weird life cycle. They're not holometabolists, they're not hemimetabolists. They're kind of this weird mixture of both. And they have like pupa that walk around, and so they're this wild like species that's kind of on that cusp there. And I think they're really cool. It's just um, having the work on them.
And from the chat, this is distinct from aphids, right? So these are still
Yes, they're different from aphids.
okay.
They're actually like an aphid would be big compared to that.
Okay. One thing that you've mentioned too, Josh, throughout the night is that y'all have been doing genome sequencing and genome assembly. One thing that we
Yeah,
always what we talk about here on stream is every Friday we do a new species of the week that's been published. And some of
yeah,
them have genome sequence, other ones is just this mitochondrial region that's sequenced. Can you just
yeah.
kind of pull back the curtain for folks on how difficult it is actually to do a genome sequence of a new animal or even of one that's still in the
It's
lab?
it's not difficult anymore. So like wildly like so if you have one fruit fly, you can probably sequence its genome. So it's better to have more like a bigger organism. Like say if you find a new raccoon species, like to sequence the genome of that, if you have the money to do it, like maybe like $5,000 to $10,000, and like someone who can devote some time to it, it's not hard anymore. Like the sequencing technology has gotten so like deep and advanced, and then there's programs to like assemble them, and then if you could, then there's like things. Oh god, to help assemble it into chromosomes now. Like you can, I mean, like it took what $1 billion in like 15 years to do the first human one or something like that. You could effectively get a lower level quality genome, like high quality genome for a new species done for 10, 15 grand. And so it's not hard anymore. And it's mainly like if you have a good, but nowadays it's no longer the like sequencing a new genome of organism that'll get you a genome report in some journal. It's you got to have a reason for like sequencing it, like maybe to do comparative stuff among like 400 to 500 different other insect species. So it's really changed a lot.
So the difficulty is just having basically the money or justification to do it now versus like actually
Yeah,
running the experiment.
yeah, yeah, and having a good like question to ask for it. And so like there's actually a few different groups. Like there's like a Darwin Tree of Life. There's a European genome where their goal is to sequence everything in Europe and the UK in those areas. We just don't have an equivalent really in the US. But it's like there's groups that are trying to sequence the genomes for every single mosquito species. There's ones that are like trying to do that sort of thing. It's I will say someone says it's like crazy expensive to do a genome. That's for a new organism. If you already have a good quality genome for an organism, it's a lot cheaper to do it. You can do it for a couple $100 sometimes.
So there it's like you're comparing to some like you have this fruit fly and you just want to
Yeah.
see how it's different from the existing genome, and so that's just
Bingo.
a few
And
hundred.
like and so you have that scaffold to map it to, and you don't have to reassemble it. And so that makes it a lot cheaper and easier to do. And so if you like that's just a resequencing the genome. And so it's like that's why like you can probably do a good quality human one for maybe $1,000 now.
Wow, compared to all the billions that it took.
Yeah,
Yeah, it's quite incredible. It's still, for folks at home, it's still relatively expensive to, us at home watching, but compared to
yeah,
what it used to be, right, it's just a
yeah,
drop in the
yeah.
bucket.
Yeah, I mean, in every year, like they have these large scale human genomes that have been sequenced. Like, oh, like you want to understand, trying to think of a disease, a specific one, like heart disease. So they'll have like hundreds of thousands of genomes sequenced, and then you say, oh, who are the people who got heart disease? And then you do these comparative like genomics for that, and you start understanding some of the underlying factors associated with that. So, or like specific types of breast cancer, or let's say lupus, like use that one as one. And so then you can start understanding what is potentially underlying those.
Yeah, that's really, really cool. Thank you, Josh. As
Yeah,
we round out for the evening,
yeah,
one thing that I really admire about what you do is too that you put a lot of time into science outreach and just like not
yeah,
just training undergraduates, but even guys, Josh does outreach at zoos as well and teaching high school students. Could you comment a little bit on just what you do in terms of science outreach and why is that important? And I guess why is it also important in part to even just talk to a broad audience here on this show? Like why is that an important thing for a scientist to do and why is it important to you?
yeah. It's just very critical because the entire goal of most scientific research is to improve like scientific like understanding, education, literacy. And so you're not just trying to like do studies to impress all your colleagues. You're also trying to do ideas to advance like just the general like kind of society, science understanding, that sort of thing. And so you really want to go and kind of do outreach that range all the way from elementary schools all the way to the general public, and try to make sure people know because like it's like there's a lot of parts of science that it's a lot of it isn't always necessarily an opinion. And so it's almost never an opinion. Whatever's correct will eventually percolate its way to the top. And so at some point. So it's just making sure that people understand like oh like when we're doing science, it's we don't there's not really an agenda at all. Like maybe individually there might be an agenda for some people, but in the grand scheme of things long term, science, if you do something, you figure out something and it's wrong, someone else will find the opposite and correct it. And then kind of keep moving along to understanding. And so the idea is that making sure people kind of understand that. And so that's really like, and it's not just like your other colleagues, undergraduates, that sort of thing. It is that you should teach undergrad classes and do a good job, but then high school and that sort of stuff. I will say, you throw that out there. In the past couple of years, I have not done as much outreach as I used to, but it's only because I've gotten so busy that I don't have time. Like having a full lab with teaching 3 classes and that sort of thing. It's once all that comes on top of each other, it's like right now during the summer, it's like I have like a 50-hour a week job just running my research lab. And then on August 25th, I'm getting 2 classes dropped on me to teach as well. And so then that adds like 12 more hours a week. So it's like you have a full-time job and then you get teaching like on top of it as well. And so that's just usually how life goes, but that's how it is to be like in academic research.
And a family on top of that too. So
Yeah,
in addition to having all the work stuff, then you also have like
yeah,
time at home too. That yeah.
yeah.
So
I mean, yeah. The one thing I will say, I really never thought like looking back into like my 20s or even like when I was in college that someone would really effectively pay me a livable salary to go play with bugs every day, but they do. So I really, in the greatest, and I 9 times out of 10, like my schedule is like as a scientist, it's fluid, but it's also dependent on the ones. One of the reasons I was easily able to be here so late, I was pulling samples out like at 8:30, right before this was happening. I knew I would be doing that today, and so there's times when I'm pulling out samples that late, other times when I'm not, and so like then tomorrow I may not go in the lab until noon, and particularly if no one needs me in the lab, I may stay home and write some days. So it's this kind of weird thing of like you're working a lot, but then like there's a complete fluidity in the schedule some days, other than when you have to teach where there's actual like class times. Like the research kind of decides the timing.
yeah,
So yeah.
awesome. Well, Josh, thank you. Is there anything that you would like to, any parting words of wisdom for the folks at home? Everyone's saying thank you messages for spending your time with us this evening and answering questions and talking about your research.
No, I mean, just the biggest thing is that kind of what I said before. Like, science really doesn't care like what you think. There's always eventually a right answer, and it will get to that eventually. And that's how science works. Like papers, like people, like there's conflicting answers in papers, but then eventually one of them is usually right. And so, and that always kind of comes to the top eventually, and that is just how the process is. It doesn't mean it's wrong. It just means that stuff that was not correct eventually gets corrected, and that's how the process of like research and scientific education should work.
It's just learning and discovery and changing
Bingo.
your answer along the way to fit the new discovery.
Yep.
Yep.
Bingo.
Awesome. Well, Josh, thank you so, so much for your time tonight and for joining us. And if you ever want to come back to talk about a new paper or anything at all, we'd love to have you.
Yeah, at some point. Yeah, I would love to come back and chat.
Thank you, Josh. Have a great
Yeah.
evening, and we'll see you
Science
soon.
is for everyone, all
All right. See you
backgrounds, all
later.
ages,
Thank
all
you.
genders,
Bye.
and all walks of life deserve an education. Come be one of us.